Hello dear students, in today's video we are going to discuss about what are the indicators. What do you mean by indicators? Suppose if you are driving a car, suppose if you want to take right turn, you will put right indicator. If you want to turn left, you will put left indicator, or brake. For all those indicators are there. For what are the uses of this particular thing? So it will indicate that you are going to turn either right or left. So in today's lesson, we are going to discuss about indicators. These are, we can define indicators are the substances whose color or odor changes once they come in contact with acidic or basic media or substances. So you might have noticed one thing. Suppose when you take some curries, suppose if you want to this particular stain of this particular curry, if it falls on your shirt, white shirt, so it will take some what yellow color. So if you wash this particular cloth with soap, you will see that you will notice that the yellow stain of the curry that will slowly changes to somewhat reddish brown. So, what is the reason behind it? In turmeric, especially in curries, subjects we use turmeric powder. So, turmeric is a natural indicator. So, turmeric has got yellow color, is it? So, the yellow color stain, when suppose some curry which is made up of, which is mixed of this turmeric powder. So, once it falls on your white shirt, what will happen? The white shirt, its color will change to yellow. And similarly, you can see this yellow color stain that will remain as it is. So when you wash, what will happen? You are applying soap. In the previous video, we already discussed that soap is uh, what type of substance? Acidic or basic? Yes, basic substance. So once soap comes in contact with this yellow color stain, so what will happen? This yellow color stain will turn somewhat to reddish brown color. So we can understand that soap is basic in nature. So once again you rinse this particular stain with a large amount of water, what will happen? The content of soap will get washed away and the yellow color will start appearing again. So suppose when you discuss about indicators, indicators we can divide mainly into three broad categories. One are called natural indicators. These are called natural indicators. Example: this turmeric powder, then leaves of hydrangea, this red cabbage juice. These are examples of natural indicators, especially litmus. So, of which we studied about 5 classification last year, which planted is under that Thallophyta. So, lichen is a small plant which belongs to the family Thallophyta or particular division Thallophyta. So, what happens? Its juice, we are extracting a juice from this particular lichens, and that we are uh, imprinting onto filter paper, so that we are getting litmus paper. So it is available in both red as well as blue color. So we have already discussed about in lower classes. And the next type of indicators are known as synthetic indicators. Synthetic indicators means these are artificially prepared in the laboratory. And the last one is called olfactory indicators. These are also natural indicators, but olfactory indicators, which means the indicators that are uh, changing their smell once it comes in contact with acidic or basic substances. Certain substances will retain that smell, but certain substances will lose its smell. Suppose we are taking two samples. One sample we are taking, that is HCl. So let us see what happens. HCl, when comes in contact with the natural indicator turmeric, there will be no change. So once it comes with red litmus paper, what will happen? The color will remain as it is. Once it comes in when it comes in contact with blue litmus paper, what happens? The blue color litmus paper will now change to red. So which indicates that the given sample is acidic. You copy this particular table to your notebook very nicely, and don't forget this. It's very important. So this is how we are going to test whether the given substance is either acidic or basic. So we cannot test in by testing it. So it is not often uh, uh, required, which is not at all specified. And similarly, we can see. So when you take the example, say synthetic indicators, we can see there are two types of synthetic indicators. And the synthetic indicators are of two types: methylene orange or methyl orange, then phenolphthalein. These are the two synthetic indicators which we have got in the laboratory. Then olfactory indicators of the third category, in that vanilla and clove. These two are the olfactory indicators. See what happens when these synthetic indicators, especially methyl orange, which is used. So when methyl orange, when it comes in contact with acidic media, it will change its color, somewhat orange color. It will change its color to reddish color. And in the basic medium, when it is exposed to basic medium, what will happen? Its color will change to yellow. Then phenolphthalein. If you take phenolphthalein indicator, suppose say 5 few ml of this particular HCl and 1 drop of this particular phenolphthalein. If you add 1 drop of phenolphthalein to this particular HCl hydrochloric acid, what will happen? It will remain colorless. It will not turn its color, phenolphthalein. Suppose if you take say 2 or 3 ml NaOH, sodium hydroxide, and pour a small amount of phenolphthalein into it, what will happen? Its color will now turn to pink. So we can understand the given substance is basic in nature. So at last we can go discuss about olfactory indicators. Olfactory means what? Olfactory indicators, these are related to smell. Olfactory means the sense of smell. So vanilla, suppose if you take a few ml of HCl and add few drops of vanilla essence to acid, so the smell of vanilla essence will increase. The similarly we can say clove oil or long oil. So if you mix some amount of acid with this particular clove oil, the smell of the clove oil will increase, so that we can understand the given substance is acidic in nature. And similarly, if you take sodium hydroxide, an example sodium hydroxide, and if you pour a small amount of vanilla essence to this particular sodium hydroxide, so what will happen? You will not be able to detect the smell of this vanilla essence. And similarly, what happens if you add a small amount of sodium hydroxide to this clove oil, the same thing happens again. So we will not be able to detect the smell of clove oil. And once we miss this particular thing about litmus paper, suppose what happens when sodium hydroxide, that's a base, we already seen the definitions which are very important, you revise that again. And what happens when red litmus comes in contact with a basic substance? It will change its color to blue. Remember that when a red litmus, if you take a red litmus paper and when it comes in contact with any basic substance, say soap, so what will happen? Or sodium hydroxide solution, what will happen? Its color will change to blue, which means that the given substance is basic in nature. So what about the blue litmus paper? The blue litmus paper won't change its color and it will remain as it is. So we can understand the given substance accordingly using these different indicators. Once more we can see what we mean by indicators. Indicators are the substances whose color or odor changes once it comes in contact with any acidic or basic medium. So there are indicators, there are three types: natural indicators, then synthetic indicators, and olfactory indicators. So especially when you take natural indicators, turmeric, vanilla, this particular hydrangea, juice of hydrangea leaf, this particular flower, then petals of this particular red cabbage leaves. These are all natural indicators. And similarly, another natural indicator is what? Lichens, especially lichens. When you extract the juice of this particular lichens, it will be somewhat purple color. So these are used for making litmus paper. This purple color dye, actually, this what we are extracting, this juice of lichen. So which is purple in color. So we are making it also red and blue litmus paper. And the next one is synthetic indicators, which are actually a mix of methylene orange, methyl orange, or phenolphthalein. These are the two synthetic indicators. The next one is olfactory indicators. Olfactory indicators means what? The substances which smell changes once it comes in contact with any acidic or basic substances. One more example is what? Extract of onion. So this you can test at your home. It just makes chop some onion and it just mix it with some amount of some cloth and then you test it with vinegar which is available in your uh, kitchen, and similarly you can test it even with the soap also. So in acidic medium what happened, the smell will retain. Basic medium, the smell will not be there. These are the major three categories of indicators. Especially this table is very important. First of all, you may find it a little bit difficult, but try to remember these are the results. This particular table you draw neatly in your notebook, it will be useful later. Okay, I hope you understood this particular lesson. Those who find it difficult, just revise and watch this particular video again. Okay, stay well and stay safe. Okay, study well. Thank you.